Hey everyone, welcome to BioBusters. In this video, we'll cover everything you need to know for Subtopic 1.5, the origin of cells, of Topic 1, which is cell biology of the IB Biology HL curriculum. I'll begin by introducing two important concepts, abiogenesis and biogenesis. We'll then look at Louis Pasteur, the Miller-Urey experiment, and the four conditions needed for life to emerge and persist. Lastly, we'll look at what an endosymbiont is, the endosymbiotic theory, and the evidence that led to it. Let's get started. Abiogenesis is a theory that living cells arose from non-living matter. Currently, living cells cannot arise independently by abiogenesis because of a concept called biogenesis. Biogenesis states that cells can only be formed by division of pre-existing cells. It's a principle that living things can only arise from other living things by reproduction, not spontaneous generation. Louis Pasteur, a French microbiologist, gave evidence to support this principle as he disproved the theory of spontaneous generation. In his experiment, he had 3 broths stored in swan neck ducts, which blocked the passage of external dust particles. His first step was to sterilize the broths. To do this, he boiled them, killing any microorganisms present in the growth medium. In the first one, he did not break the stem. The result was a clear broth showing that no microbes had formed or become trapped. In the second one, the stem was broken. The result was a cloudy broth, showing that microbes from the air went to the flask, grew, and multiplied. Lastly, the third flask was tilted. The result was a cloudy broth, as it was exposed to microbes in the curve of the swan neck, showing that microbes grew. So this experiment explains how new cells come from older cells. But where did the first cell come from? To find this, Stanley Miller and Harold Urey conducted an experiment, the Miller-Urey experiment, and recreated the conditions of early Earth in a closed system. It had low oxygen levels, high radiation levels, high temperatures, and electrical storms. To do this, they boiled water to vapor, mixed it with hydrogen gas, methane gas, and ammonia. Then they exposed the mixture to high electrical discharge and let it cool. After 1 week, simple amino acids and complex oily hydrocarbons were found in the reaction mixture. This proved that the non-living synthesis of simple organic molecules was possible. The results of this experiment led scientists to believe that there are 4 conditions needed for life to emerge and persist. 1. Simple organic molecules, such as fatty acids, amino acids, and carbohydrates, must be formed from primordial inorganic molecules. 2. Larger organic molecules, such as RNA, DNA, and phospholipids, must be made from simpler molecules. 3. Organisms reproduce, so replication of nucleic acids must be possible. And 4. Self-contained structures, for example membranes, are necessary to maintain set conditions for reactions. Another important theory on the origin of cells is the endosymbiotic theory. But first, what is an endosymbiont? It's a cell which lives inside another cell with mutual benefit. The endosymbiotic theory explains the origin of eukaryotic cells. It states that eukaryotic cells evolved from prokaryotes that were engulfed by phagocytosis and remained undigested. Cells that did aerobic respiration evolved into mitochondria, and those that did photosynthesis evolved into chloroplasts. This is believed because both mitochondria and chloroplasts have many characteristics seen in cells. For instance, double membranes, DNA, single chromosomes, 70S ribosomes, and they also divide using binary fission. And that's it for Subtopic 1.5 of the IB Biology curriculum. Thanks for watching!